Look to the person to your left. Now look to the person to your right. One of you three will likely get Alzheimer's disease by the time you're 80. It'll start with dementia, or it'll start with memory loss, and it'll proceed to dementia and eventually death. This is a catastrophe. It's going to affect our healthcare system for the next few decades. And more importantly, it's going to rob us of those that we care about. We need to find new ways to address this disease and new creative ways to figure out how to stop it. So my lab at the NICEF Research Laboratory is trying to use stem cell biology in a unique way to try to address this disease. But Alzheimer's is actually probably many diseases. And like cancer, each of these types may have different molecular root causes. The root cause of my Alzheimer's or my dementia may be very different from your dementia, and therefore the drugs that we use to treat them, if one works for me, it may not work for you. So we need to come up with personalized therapies that can treat each type of these dementias. So currently, a physician knows, there's basically two ways that a physician knows whether or not you have Alzheimer's disease. In the first case, there are a small number of cases where a single gene mutation guarantees that you can get the disease, and usually by your mid-40s. But in the vast majority of cases, those of us that get it in our 60s, 70s, and 80s, a pathologist has to wait until autopsy in order to understand whether or not you definitively had Alzheimer's or a different type of dementia. And the disease actually probably starts up to 25 years before you show clinical signs of dementia to your doctor. Trying to go back. There. So for example, a pathologist would look for things like amyloid plaques, the tangles that arise inside the neurons in the brain, reduced brain volume. All of these are physical manifestations of the disease. There are imaging technologies that are available today that allow us to track the progress of this disease, but usually very late in the disease process compared to this 25 years from the start. This has been a major limitation for us understanding how to actually understand the beginning of the disease. And currently, there are no drugs that are approved to treat Alzheimer's disease or actually prevent it. And we're still left to wonder what went wrong. But to be sure, we've understood a lot about the end of this disease, but trying to understand its beginning or how your cells are actually experiencing the disease has been very difficult. And we can't go into the brain of a living Alzheimer's patient and ask for their living brain tissue in order to study it in the lab. We need to find a better way to get access to this disease in the lab. So as a scientist interested in studying Alzheimer's and in finding a cure for Alzheimer's, I want to understand the beginning and how it progresses to the end. But as a stem cell biologist, I also have access to I have the opportunity to use living human cells. I have the opportunity to, in the lab, study how these cells develop, how they progress from healthy into a disease state. And I can do this in the laboratory. This is critical for us trying to find the earliest possible point to treat this disease. So for example, using a Nobel Prize winning technology, we can take adult human cells and turn back the clock and recreate stem cells from those cells. And these cells have the property of pluripotency, meaning they can develop into any cell type in the body, so heart tissue, brain tissue, the beta cells of the pancreas, bone tissue, any cell type. Most people think of stem cells as a way of generating replacement tissue for dead and dying organs in the body. But I'm more interested in how the disease actually gets started in the first place. So in my lab, we are generating human models for these diseases in order to understand how these diseases work, and we're using them to test drugs and to find better diagnostics. We've also automated the process of deriving stem cell lines, in order to look at large populations of people that actually have this disease. So for example, I can take a skin cell from a patient with Alzheimer's disease, and I can turn that skin cell into a stem cell. And then by mimicking the processes of development, I can turn those stem cells into the brain cells that are affected by the disease. This allows me to have a living window into the disease as it progresses. We've been doing this from patients that have the genetic forms of the disease, the kind that guarantees that you will get it. And we've been able to see the key indicators of the disease, things like the plaques and the tangles start to develop in the petri dish. And we can see this without having to wait years and years as this disease progresses in a patient. But what about the other 95% of people that get Alzheimer's disease but don't have these mutations? How do we understand their disease? This is currently a very difficult question because it's hard for us to predict who is actually going to progress into Alzheimer's disease. Just because you're forgetful doesn't actually mean you're going to get Alzheimer's disease. But ideally, we'd use these same techniques to try to understand these forms of the disease and try to understand how this 95% of people actually get the disease. So when developing models of this disease, we need to start with the controls. We need to understand how, we need to find cases of the disease where we definitively know that these people died from Alzheimer's disease and not a different kind of dementia. This is something that we can only currently do after death, definitively. But to address this problem, my lab has been able to recover living cells from cadaver tissue from people who have died from Alzheimer's disease. And we've been able to recreate stem cell lines from those cells. And these stem cells behave as the other stem cells do. They have the property of pluripotency. They can make all the different cell types. But we can go a step further. We can recreate the brain tissue from these people that died of this disease. We can recreate the neurons that were affected by this disease from these stem cells. What you're seeing in this movie is a network of neurons recreated from the stem cells from a patient who had already died from Alzheimer's disease. These neurons form connections and synapses, and they have spontaneous activity, properties that are similar to those found in the brain. It's these neural network connections that are the building blocks of learning and memory, and they are the target of this disease. In order to understand how to develop therapies for this disease, we need to understand how these connections are targeted by the disease and how the cells eventually die. And we're starting to do this now. What's so exciting about using stem cells from the deceased is that it gives us a new starting point to try to understand the beginning of the disease. And instead of trying to answer what went wrong only at the end of the disease, we have the opportunity to replay Alzheimer's disease over and over and over again in the dish to understand the beginning of the disease. And with these new insights, we should be able to develop more effective therapies for Alzheimer's disease. This is something that we can't currently do now. And intervening at the beginning is when it would matter the most. This is where Alzheimer's disease lives, and this is where we could probably go to find the beginning of the disease and how it progresses more effectively, and maybe find a cure. Thank you.